Hello everyone, I'm Evariste and thank you for watching this presentation entitled Ecotoxicity of Graphene-based Material toward the diatom-bacteria biofilm. Nanomaterials are composed of at least 50% of particles that possess one or more external dimension in the size range comprised between 1 and 100 nanometers. And of course, this definition includes carbon-based nanomaterials such as the graphene, that is a single layer of carbon constituting the basic element of any carbon allotrope. And due to its properties, there is a growing interest for these nanomaterials for the development of new application in many areas. Graphene-based nanomaterials are mainly made from graphite that can be exfoliated and oxidized to produce graphene oxide. And due to the presence of oxygen-containing function at its surface, it can be functionalized. And due to its high stability into liquid, it's used also for polymer reinforcement. This graphene oxide can be reduced into reduced graphene oxide, and this one possesses good properties of conductivity that are used mainly to produce electrochemical sensors. And these two materials are willing to be mass-produced because they are cheap and easy to produce, contrary to high-quality graphene that possess best performances, but is actually too expensive to produce and into limited quantities. As the production of these materials is expected to increase, their release into the environment is also expected to increase. So there is a need to perform ecotoxicity assessment of these materials to establish risk mitigation strategies when needed, and this in order to contribute to the sustainable development of these nanotechnologies. Concerning the ecotoxicity of these materials, most of the literature available target on the effect toward bacterial energy, and common toxicological mechanisms were highlighted. For example, the first one consists in physical damages that is leading to membrane impairment, and also to the rust production that is leading to growth inhibition of the organisms. However, all of these studies were performed using single species based assays that is not relevant for environmental conditions. In the environment, multiple organisms are interacting. This is the case, for example, for biofilms that are microorganisms embedded in EPS. And this lifestyle provides different properties compared to free-living organisms. For example, it provides enhanced resource capture and also survival to antimicrobials. For our study, we choose the model of biofilm composed of diatom associated to a bacterial consortium. So the diatom are producing EPS that are providing trophic resources for bacteria that produce nutrients that are also beneficial for the diatom. And very few work are devoted to study the responses of biofilm exposed to graphene-based nanomaterials. So this is what we expected to do with our study to fill the gap remaining. For this purpose, we assessed the effect of a commercial form of graphene oxide and reduced graphene oxide toward a biofilm composed of the diatom niche palea associated to bacterial consortia. So the organisms were sequentially introduced into flask before performing 2 days or 6 days of exposure. At each sampling, we analyzed different physiological endpoints for the diatom using flow cytometry. Same for bacterial growth, and also some function were analyzed using biolog ecoplate. And also the community composition was monitored using 16S sequencing. Concerning the growth parameters of the diatom, we observed that the growth was significantly stimulated when exposed to graphene-based nanomaterials as soon as 2 days of exposure. The maximum growth rate was reached after 2 days in graphene-based containing condition. And this maximum of growth was reached into the control group after 6 days of exposure. And at the end of the exposure, we did not observe any differences between the growth rate into the control and the exposed condition. And we think that it's mainly due to silica limitation in our system. Concerning the effect on the diatom physiological parameters, I choose to show you 2 main results. The first one concerns the chlorophyll content of the diatom, And this time, following 2 days of exposure, we did not observe any change in these parameters in any condition. However, after 6 days of exposure, we observed an increase in chlorophyll content following exposure to high GO concentrations. This indicates that phototrophic metabolism of the diatom is increased under this condition. While following RGO exposure, we can note a slight decrease of the chlorophyll content. The second physiological parameter is the lipid content of the diatom. And this time, following 2 days of exposure, we observed an increase in the lipid content following exposure to RGO. And this effect was also noticed following 6 days of exposure. And this indicates this time that the diatom is increasing its heterotrophic metabolism following RGO exposure. And this also constitutes an indicator of stress that was observed in situ into Niche Palea, where organisms from polluted areas possessed higher lipid content. Now let's switch on the bacterial compartment of the biofilm. And the first parameter that we monitored was the growth of the bacterial consortium. Here, following 2 days of exposure, we observed a dose-dependent bacterial growth inhibition following GO exposure, while only slight effects were observed following RGO exposure. And these effects were also observed following 6 days of exposure. So from the result of GO exposure, we can determine EC50. And the interesting point is that the EC50 is increasing over time, so it indicates a kind of recovery. To assess the effect on carbon source consumption, we used Biolog EcoPlate. So with this plate, 31 different carbon sources can be assessed. The carbon source is associated to redox dye that can be oxidized with bacterial metabolic activities. And this is producing a purple stain that can be analyzed and read using plate reader. Considering the overall metabolic activities, following 2 days of exposure, we noticed only a slight decrease of carbon metabolism following exposure to graphene oxide at 10 mg per liter. But if you remember, we had a strong growth inhibition under this condition. So if you consider the metabolic activity per bacteria, this time you observe that the bacterial metabolism is increased in gel-containing condition, and this is acting like a compensation mechanism. And after 6 days of exposure, this time we observed no significant differences between the different tested conditions, and we only noticed an overall decrease of the activity with the biofilm maturation. Now, if we focus on the result from the 16S sequencing, we observe that following 2 days of exposure, the diversity and the community structure is not affected by GBMS. And this is surprising because we had strong growth inhibition while the community was not affected. So this is suggesting that all the species are affected in the same way. So there is no resistance associated to specific species after 2 days of exposure. However, after 6 days of exposure, we can note an increase of the Shannon index following exposure to GO at 10 mg per liter. And the community structure was also significantly different in this condition compared to the other ones. When analyzing the bacterial community composition of our biofilm, we observe that it is mainly composed of bacteria from the phyla Bacteroidota and also Proteobacteria. And it is interesting to note that the ratio between the two phyla is evolving over time with the maturation of the biofilm. And now if we have a look, following 6 days of exposure to GO at high concentration, we observe an increase of the Proteobacteria relative abundance. We determine also the discriminant OTUs, which relative abundance were significantly different from the one found in the control group. And through the use of DSEC, we found 4 OTUs that were differentially abundant following 6 days of exposure to graphene oxide at 10 mg per liter. And the interesting point is that the 2 first ones are increasing over time in the control group, while their increase is much more important following GO exposure. And the 2 last ones are presenting a different pattern, while their abundance is decreasing over time in the control group, while it's less decreased following GO exposure. And these 4 OTUs are belonging to the phylum Proteobacteria. That might explain the increase of their relative abundance in this condition. And the interesting point is that these bacteria were shown to be involved in the degradation of contaminants. To summarize all of these results, so first with the diatom compartment, we noticed growth stimulation induced by graphene-based material exposure. We also observed that the exposure was able to influence the diatom metabolism with a greater impact of RGO. For the bacterial compartment, we observed a growth inhibition mainly induced by GO exposure and also an increase of metabolism. And the community structure was also affected by graphene oxide exposure. Concerning the interaction between the 2 compartments, we can note that the diatom and the bacteria are directly affected by graphene exposure, and also that for the diatom, that the exposure to a GBMS was leading to an increase of EPS secretion that can be used for by bacterial communities to produce nutrients and that are beneficial for the diatom growth. However, we still don't know if the EPS composition is affected, and this could have an indirect effect on bacterial community structure. We also don't know if the biochemical composition of the biofilm is affected, and this could have consequences for higher-tier organisms feeding on this biofilm. And to have some answer to this question, I invite you to read our last article published in Carbon. And thank you very much for watching, and take care.